Disclaimer, disclaimer, disclaimer. We humans may see ourselves as independent life forms, liberated from animal ignorance, able to attain the greatest heights of intellect through powers of logic and reason. And when engaged in this pursuit, we do quite well. But much of who we are and how we live is still under the direct control of a higher power. For the true world of man is the world of the brain, and within the world of the man's brain, there lives two distinct creatures. There is the sentient mind of self, that with which we think, scheme, plan, and dream. And then there is that other creature, the animal control brain, the controller that manipulates us into eating, urges us into lovemaking, reminds us to care for our young, and commands us to seek safety and security. If we forget to eat for a few days, it is not the sentient knowledge of needing nutrients that nudges us into nibbling, but painful pangs of stomach sensations brought on by the animal control brain that lightly punishes us into eating. And once we have eaten, we're not intellectually satisfied by our self-nourishing act. No, we are injected with opiates, serotonin, oxytocin, dopamine by the animal brain controller as reward for following its commands. The same is true of sex, of love, of safety, of the pleasing appearance of order. The controller is actively punishing and rewarding the sentient mind, manipulating mind and body in acts of basic animal survival. And as it asserts its dominance over the will of man, again and again we submit to its ultimate authority, fanatically, without question. We obey, not because it is in our best interest to do so, but because our best interest was determined by our caretaker and tormentor long before man existed. That it bears great similarity to the myths created by man to explain a controlling supernatural force greater than himself is not a coincidence of terms, but may be the explanation for the coincidences of myths throughout the ages. The one thing that allows us to escape the animal control brain's dogmatic directions, our intelligence. We can have sex for the sensation alone, not just procreation. We eat foods that release the greatest rewards and get extra oxytocin love hormone delivered by music or watching movies. We have learned much about the brain itself, giving us an ever-increasing advantage over the controller and our understanding of how much we require its control. And at the end of each day, when the eyes close and the world in which we live is once again reduced to the inner skull walls, we can be thankful for the knowledge we have gained and the perspective we have learned by listening to This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kirsten and Blair.
Good science to you, Justin. What's it like sitting there in the big chair?
I feel powerful. There's so many buttons. There's a red button that's flashing here, right here. I'm totally tempted to push it, but I'm just
But we
barely.
all know from Ren and Stimpy, don't push the red button. Don't push the shiny red button. Oh, everyone, welcome, welcome, welcome to another episode of This Week in Science. Blair and I are sitting here in my home office. There's Blair's arm. We have Blair's arm in the shot. But Justin is at the TWiT studios. He is experiencing the cavernous wonders of the new studios for himself. And it's exciting to have somebody there. I'm sure Brian likes the company.
Yeah, no, it's a fun place to hang out. This is it's pretty. There's all sorts of exciting things that go on here behind the scenes that none of you ever get to see.
That's right.
I witnessed a shanking.
The magic behind the curtains. That's right. A shanking? I don't know if I want to even hear that story. So we're going to get into the science, right? Because there are lots of science stories for the hour. Today, I've got news about your bee-like brain, or how your brain is like a bee, some disease treatment, world robot domination, and the spawning of life's diversity.
Wow.
Yeah. What did you bring?
I've got viral bats, mad cows, overthinking God, and how not to discover the G-spot.
Interesting. I saw that study. I kind of expected that you would bring it. So I was like, yeah, I think that's a Justin story. Blair, what did you bring today?
I brought a story about meerkats and how the losers in the colonies are actually smarter. Sound familiar?
Smart losers. No.
Nerds.
Nerds. Oh, you. There we go.
See what I did there?
I see
Okay.
I see what you did there. All right, let's jump into it. How about the spawning of diversity? All from just a few exploding stars. Not little exploding stars, but actually kind of big exploding stars, actually. Supernova. So we know that supernova explode, and they are these massive sources for galactic cosmic rays, which are super high energy particles. So protons, gamma rays, neutrons, high energy electrons, all sorts of particles that just shoot out at near the speed of light from the supernovas. And our planet has been moving in its journey around, in its orbit around the center of the Milky Way galaxy for billions of years. And we know, or at least hundreds of millions of years, but billions of years, basically, it's been moving around and around and around and around. And as it moves, orbiting the center of the galaxy, our planet has encountered supernova, or the stuff that gets shot out of supernovas. And so it's possible that our planet has been impacted significantly throughout history by the cosmic rays and other energetic particles that are emitted from supernovas. So a researcher, Henrik Svensmark of the Technical University of Denmark, has done an analysis going back 500 million years, looking at geological and astronomical data, looking at how close our sun has come throughout history to various supernova events. And so whether it's closer, would have a more direct impact from cosmic rays, or further away would have a lesser impact. The interesting thing from this study is looking at the movement of the continents, so the geological data. The movement of the continents is known to have affected biodiversity over the years. But when he took the movement of the continents out of the equation, he found that there was still a very significant effect on biodiversity that correlates, I'm not saying causation here, but correlates to supernovas, and correlates in a fascinating way. And there's a graph that shows from this study that shows the biodiversity, marine invertebrate biodiversity. He was looking at these shelled creatures and lots of carbon-containing organisms, relative supernova rate, and geological time over millions of years. So going back, like I said, over 500 million years. And biodiversity, the relative supernova rate are really closely matched.
And you're saying closely matched. I'm looking at a chart that that's put up there of the overlay of the two, and it's amazing.
Not exact, but
it's amazingly close, though. I mean, not exact in moment for moment, but if you look over the scale of the thing, is when it's all going up, everything's going up. When it's going down, everything's going down. It's it looks pretty good.
Right. So
It may be trailing. One might be
correlation.
trailing the other, but yeah.
One, and that's exactly it. One is slightly trailing the other. And so there's an intervening step here. So we're not talking cosmic rays leading to mutations.
Really? Because that's what I would think.
Not causing mutations directly, affect just directly mutating DNA and causing animals to spontaneously evolve.
No, but an unspontaneous. I mean, then it's not the spontaneous mutation. Really, we're talking about. We're like these are minute mutations that then over generations become more and more expressed.
There could be that kind of mutation going on, but these particles, cosmic rays, are so energetic that they probably just pass right through us. Very, very rarely actually interacting and causing damage or changing DNA. It's possible that there is a role there, but the stronger role is actually through climate. So we know that cosmic rays influence atmospheric activity. And so increases in supernova activity could lead to increases in cloud cover, leading to at certain levels of the atmosphere. Also could potentially affect carbon processing within the atmosphere, and lead to a colder climate and a glaciation period. And then the lag that you were mentioning that you see in that graph, the lag is actually the difference between, okay, supernova turns on, we have a lot of supernova activity, and then the climate changes, then biodiversity increases, supernova activity decreases, the climate starts to warm up again, and then biodiversity starts to decrease. So it's a very interesting idea. I don't know necessarily whether, all of the arguments are accurate, but it's an interesting observation, looking at the data and trying to put the story together to, figure out, okay, did supernovas, we know these exploding stars, they have seeded the universe with the heavy elements and the materials that our planets are made from, that our own star is made from. So they've not only seeded the universe, our galaxy, with the materials for creating the planets and our Earth, which could support life, but maybe actually are responsible for increasing the diversity of life. Supernovas. Thanks for everything.
Or there's a yet to be discovered mystery unknown force that is causing diversity and supernovas simultaneously.
But then you wouldn't have seen the lag.
Well, no, but the lag isn't, according to the graph, it's sometimes a lag and sometimes a precursory event. It actually switches sides a couple times. Yeah. And so, maybe it's one event, depending on which direction it's coming from, that, I don't know. But then some of these times are sort of approximations anyway. It looks like based on the way that they've collected data, it looks like it's probably a perfect match. It's just some of the datings of when this, that, or the other happened are being slightly off. I would guess that those are much closer related even than the graph indicates, because of the way they trail, and even the little ebbs are represented in the other. It's, it could be a correlation, but gosh, that's a wicked accurate accident. That's like lottery number kind of accident, right? That's like, when you see that kind of a correlation, it's like, it's just a correlation at this point, but it really looks good. We just don't know why it's happening, so we haven't figured out how it's linked exactly.
And now this opens up a whole new area of investigation. What kind of questions can come from this? So, how can experiments be devised that could actually test this further? Because so far it's observation, it's correlation. What can we do from here on out to actually experimentally investigate this idea?
And bombarding living creatures with gamma radiation sounds like probably the next step.
Right. Hey, Spider-Man, what's up?
Something, maybe yeast. Maybe they start with yeast just to see if they can get some sort of a change to take place. It'd be interesting. It'd be fun.
All right. Give me a story.
It's the last thing you want to find in most cases. But if you're a scientist, you are very excited to have discovered an unexpected virus reservoir. What? Yeah, this is researchers from the University of Bonn discovered that bats may be not just a common source of many viral diseases, but one of the most common sources of diseases that affect humans. Professor Dr. Christian Drosten, head of the Institute for Virology at the University of Bonn, says, "We already knew from prior studies that bats and rodents play a role as carriers of paramysoviruses." So the professor, the many varied members of this large family of virus, measles, mumps, pneumonia, and the common cold, right? Highly dangerous also, a couple of the viruses that are in this family, Hendra and Nipah viruses, which cause types of encephalitis that result in death for 1 out of 2 patients. If there are 3 patients, a game of Rochambeau determines who lives and who dies. So paramysoviruses also play a role in veterinary medicine. It is the cause of canine distemper, which I have no idea what that is, but apparently it makes dogs angry. So with the support of numerous scientific institutes in Germany and around the world, they tested a total of 9,278 animals from Europe, South America, Asia, including 86 bats, 33 rodent species. These animals live in a very large social community with millions of individuals, in some cases, reported the professor. Their close contact promotes mutual infection and provides for the greatest variety in circulating viruses. So, because they're mammals in huge populations, one virus gets in somewhere, it circulates really rapidly. Using molecular biology methods, the scientists identified which virus species are rampant in bats and rodents. According to their own estimates, they discovered more than 60 new species of the paramysovirus. Which is, for context, 60 new is about the same number as they knew existed. So basically it doubled the number of...
Doubled it, yeah.
Using computational biology methods, the scientists calculated a common evolutionary tree for the new and the known viruses. They then did deductions with mathematical methodology to determine in which host animals the viruses have most likely taken up residence during their evolutionary history. So, sort of backtracking where these viruses have been in which species over time. The analysis showed almost all of the forebears of today's paramysoviruses have existed in bats. Just as with influenza, says Professor Drosten, where we are keeping an eye on the birds as a source of new pandemic viruses, we will now have to study the bat viruses to see if they are a danger to humans. So the current data might play a useful role in early detection and prevention of epidemics, which is a major new goal of the virus research.
Yeah, so if you can get in there and find out what's circulating, what has circulated, you might be able to predict, what's going to jump out.
Right, which I believe when they backtracked SARS, it got identified as bats.
Wasn't. I don't think it was bat. It was something else.
I think it was the bats. They found there was like a meat market where the thing was sort of the origination point, and it was a place where people eat bat meat.
Yeah.
And then they found bats. I think it was SARS, unless there was another sort of pandemic-y Asian flu thing.
There's so many.
There was a few, but I'm pretty sure SARS ended up in bats.
Or started in bats, you mean?
Correct.
Not ended up started there.
I'm sorry, excuse me. Yeah, the other way around. It was originated from bats. Yeah. And it's also a very important point. If vaccination...
Civet Cat says someone in the chat room. I think it might have...
I don't know if it's cats. I mean, I'd be more than happy to blame it on cats, because that's fine.
Cats.
Well,
Go on with your other thought.
The professor makes a very important point here. If the vaccination campaigns are stopped once a virus has been eradicated, this might actually present a potential risk, which we will maybe have to rethink, because the assumption now is that there is a vast reservoir viruses that affect humans that exists in bats. So as if, for instance, you vaccinate against the measles, the mumps, or this, that, or the other, and then we've had enough people immunized and vaccinated for the last 20, 25 years. Nobody's come out with one of these for a while. We don't need to do the vaccinations anymore. Problem being, the reservoir is still out there. Our population can get reinfected by these bats at some point.
If not enough people have come into contact with the virus to actually give some kind of herd immunity, then yeah, it can be a problem. Yeah. And looking online, the origin of SARS, you're right, supports the bat as a virus source. However, back in 2003, researchers were thinking that it jumped from civet cats to humans. So probably what happens is it started off in a population of bats. Bats were brought to market. The bats and civet cats then swapped viruses and then on to humans. So, hence the story. Yeah, that's fascinating. I love the investigation of trying to find out what has happened or what will happen, what is circulating, what viruses are out there. And if it's anything like bacterial biology, where we have really no idea of all of the bacterial species that are out there because we can't culture them all in the laboratory. Some of them just don't grow in the lab environment, in the petri dish that we give them. If viruses are any similarity, if they, then we really have been underestimating the number that are out there. And so the number that you see, the doubling, 60 more viruses from doing these genetic scans. It's fascinating. Fascinating. All right, everybody, you're listening or watching you're watching Twis, listening to Twis. And right now, I think it's time for Blair's Animal Corner.
Blair's Animal Corner with Blair.
With Blair.
Yay! So today I have a story about Meerkat Society. They're a very social animal, and for those of you who don't know, they're Timon from The Lion King. That's what most people think about when they think about meerkats. But these guys...
I think of Meerkat Manor.
Well, of course, that's a good one, too. They usually have a dominant female, and then everyone else has a ranking within the society. And they found in some recent research with wild meerkats that the underdogs are the best at problem solving. The subordinate males well outperformed the higher up males in the society. So basically they took these meerkats and they took scorpions, their favorite treat. Scorpion. And
I love a good scorpion in the morning.
They put them in a clear container with an opaque lid. And they had to get through the opaque lid to get to the scorpion, which for a meerkat with a tiny little meerkat brain is very difficult to figure out because
I don't see it.
I can see it over here, but in order to get to it, I have to go here where I can't see it. So it's a
and
complicated
then I forget.
task.
What was I looking for?
Exactly. And so a lot of the higher ranking meerkats would try a little while and then give up. But the lower ranking meerkats had an amazingly high success rate. What they found was that even after showing them the same challenge over and over, they didn't seem to work out a rule or an attack plan, but they just seemed more persistent.
Mm-hmm.
Which does make perfect sense, because if you're a higher up in the society, you might get brought something, or if you're stronger, you can steal it from someone who spent all that time getting it. But if you're kind of in the outlying region of the society there and you get forgotten about and not taken care of, you have to fend for yourself. And so it would pay off. The only way you could survive, essentially, is to be more persistent and better at getting into these difficult places to find food. And so they modeled it after
maybe the kinds of places that would not be the optimal places to find food.
Exactly. So they were trying to model it kind of after when they have to go into it was a BBC News story, so they said crevasses to find the scorpions. So it was kind of a natural behavior for them, and something that maybe if you were a higher up social stature, you just wouldn't bother with. It wouldn't be worth your time.
Because you have the ladies bringing you scorpions.
Yes, or the other men. Or you could just, beat that little meerkat up to the side and take it from him too.
Ah, you got a scorpion.
Yeah.
I want
mine.
Yeah, and if you're that beta animal or even a delta animal that's on the outskirts, no muscles, not really a lot of gumption for fighting. Yeah.
Right. And so if you think about it, the only way
crafty
for them to survive and then, pass on genes, because it all comes down to that, is to be more persistent. There has to be some strength to being you, even if you're way on the outskirts of society. You have to find some way to still be able to survive and be worthy of passing on your genes. So for them, they can get through the opaque lid, apparently.
That's awesome.
Yeah.
Go little nerdy meerkats.
Yeah.
I like that. I like the idea that it correlates to the human nerds.
The football player stole my lunch, so I'm going to go to the lab and make something.
I'm going to go make some lunch. I'm going to become a food chemist. I'm going to create a what is it? The Star Trek things where they made the food. Losing the words in my brain. Replicator. Thank you. Yeah, thank you. Just losing the words in my brain. Thank you, everyone in the chat room. Yes, replicator. Replicator.
Everyone shouts at once.
I think but on the other hand, I think I just had my scorpion taken away from me.
It was such a good, tasty little scorpion, too. Yeah. So I've got a quick story, kind of related to animals in a sense, but it's more about memory than animals. So if you're talking about the way that an animal like a meerkat is going to be foraging or looking for food, there in animal behavior research is a theory called optimal foraging theory. And in optimal foraging theory, if you're hunting for scorpions and there's an area that has a lot of scorpions and there's another area that has fewer scorpions, you're going to go look in that area that has more scorpions because scorpions found per amount of time is going to be a great ratio, right? So you will forage there. You go after the scorpions in that one place until it starts taking an inordinately longer amount of time to be able to get your next scorpion. And at a certain point, according to optimal foraging theory, you will switch to the other patch of scorpions because the time that it will take you again will be more optimal in this other patch because of the number of scorpions. So optimal foraging theory has been great, shown over and over again, from hummingbirds to honeybees. And now a researcher has applied it to memory in your brain. So when you are asked to think of types of animals, just start naming types of animals. Blair, name animals.
Hippo.
And then from hippo, you go to
toucan.
Wow, you went to birds. Really? You're just gonna go.
Rhino, otter, snow leopard, tiger, lion, eagle, hawk, falcon. Okay, so we're going in
You went
categories.
into a new patch.
Yeah.
So what you heard there with Blair was that first she started off kind of with the zoo animals or maybe animals found on the African continent. But then one of the animals, the toucan, probably triggered her switch to then birds, going then to hawks and other types of birds. And so she switched categories. And the researcher in this Indiana University Bloomington study found that I think it was. Yeah. So that's where it was. They found that optimal foraging theory perfectly predicts how and when you will switch to a new patch or category of items within your brain. And so in thinking about how the brain works, it gives us some interesting insight into how things are categorized, and that our brain clusters things in categories, and it allows the brain to search more efficiently for information, particular kinds of information anyway. And this was published in Psychological Review. So I think it's a fun study. I like it a lot. Is it time for us to go to the break?
I think it is. I've got a bunch of stories ready to roll here, but they can't get into the first half of the show. The first half of the show is too full of sciencey goodness. They're going to have to wait.
All right, here we go, everybody. It is time for the second half of the show. Stay with us. We're going to have a break, and we will be back in just a few moments. I hope you keep listening. We've got World Robot Domination coming up in the second half. Dominate! Audible.com is the leading provider of audiobooks with over 100,000 different titles in a variety of genres, and Twis has found many science-based books in the Audible library. You can start a free trial today and get any audiobook download for free. All you have to do is sign up at audiblepodcast.com/Twis. Go to audiblepodcast.com/Twis right now for your free download. Get just about any book that you want. It's a great, great deal. Earlier today, I was thinking Guitar Zero by Gary Marcus. It's a great choice. It's a great choice of a book. All about adult learning. audiblepodcast.com/Twis. Free audiobook. Do it. Twis also has merchandise that you might enjoy. Head over to twis.org to purchase our 2010 Science Music Compilation CD and a World Robot Domination t-shirt. We have CDs and t-shirts for sale at our website. And Blair and I are going to be working tirelessly tomorrow to get some mailings out to those of you who have already done so. Be like your fellow minions and get yourself a CD or a t-shirt. If you're not a joiner for buying merchandise, maybe you will consider a donation instead. Twis is supported by listeners like you, and your donations pay for our hosting, bandwidth, contractors we need to hire, and lots of fun things that we try to do. We do try to do fun things occasionally, and we really appreciate any amount that you can give, $2 to $2,000 to even $20,000 or $2 million. You make this show possible, and we've made it very easy for you to donate via PayPal on our website. So you need to go to twis.org, go to the most recent show episode page that's up there. Take a listen to the show if you haven't done so already. Maybe make some comments, about the show, things you liked, things that you had questions about. Even critique us. I don't care. And go to the bottom of the page. There's some little pink buttons for donation. Just click one of those and your choice. Donate. We thank you for your support. We really couldn't do it without you.
Can't explain the things you heard with more than
heart. Our first assumptions were correct. Disprove the rest. It's hard. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
And we're back with more This Week in Science.
Science! That's right. We've got all sorts of science lined up for the second half of the show. Justin, make it happen.
I'm going straight to the G spot.
Oh, my.
After hundreds...
You teased it at the beginning of the show.
This is, I think there was forewarning, right? I didn't just go right for the G spot. I teased it a little bit, and now we're actually going to the story.
All right. It is a story.
There's an actual story here. This is a scientific story, so everybody hush up. After hundreds of years of searching, science finally finds female-friendly G-spot. For centuries, women have been reporting engorgement of the upper anterior portion of the vagina during stages of sexual excitement, despite the fact the structure of this phenomenon had not been anatomically determined by men of science, researchers. They never found it. They couldn't determine if it was actually there, despite, again, hundreds of years of people attesting to the fact that it existed. New study published in the Journal of Sexual Medicine documents that this elusive structure does exist anatomically. So no longer when somebody tells you that G-spot doesn't actually exist, it's all in your head. No, they found it. This is Adam Ostrensky, MD, PhD, of the Institute of Gynecology in St. Petersburg, Florida, conducted a stratum by stratum. What does stratum by stratum mean, Kirsten?
Layer by layer.
Layer by layer anterior vaginal wall dissection on an 83-year-old cadaver.
The cadaver wasn't 83.
It doesn't specify. It says 83-year-old cadaver. I don't know if that cadaver is a 23-year-old woman who's in the cadaver's been sitting around for 83 years or...
No, I think that the woman had died at age 83 and donated her body for science.
The dissection established the presence of the G-spot, a well-delineated sac structure located on the dorsal perineal membrane, 16.5 millimeters from the upper part of the urethral mentas, creating a 35-degree angle with the lateral side border of the urethra. Hang on, I'm just going to make a note real quick. Having 3 distinct regions, the G-spot emerged with dimension of length of 8.1 millimeter, width of 3.6 millimeters to 1.5 millimeters, and a height of 0.4 millimeters. Upon removal of the entire structure, with the adjacent margin tissues, the G-spot stretched from 8.1 to 33 millimeters. This study confirmed the anatomic existence of the G-spot, which may lead to better understanding and improvement of female sexual function, Ostrozensky concludes. This is one of the problems in science that science sometimes suffers from. The G-spot had to be discovered by dissecting an 83-year-old cadaver, right? It had to be discovered in a corpse. And in a sense, how far removed from the joy of discovering such a thing on your own could this research possibly have been? You know,
there is that, but there's also the fact that this is one study. This is a single individual. He did not do any neurophysiological studies to actually
kind of hard on a corpse.
Right, exactly. There is a lot to question about this study. I mean, not to mention the fact that the surgeon who did this particular, I have to call it an anecdote. I mean, it's an observation because it's a single data point. It's a single data point. He's a cosmetic gynecologist. So, scientifically, it's interesting. But I mean, he would be interested in the tissues, what they hold. I mean, it's the idea that this is this one particular spot and he has located it specifically.
And it could be in different places, in different locations, and different women, which is very highly my anecdotal knowledge would attest to. The thing too is that, you know, it would seem like this could have been discovered at any point previously. And I always thought the elusiveness of the nature of the G-spot was the fact that it would probably have to be discovered while somebody was being excited, and then eliminating the whole, you know, ability for a surgeon to be any way involved. It sort of eliminates that whole anatomical discovery that took place here. But you know, it's one data point. You're absolutely correct, and much more data will need to be collected, and I think hopefully under different circumstances.
Hopefully under different circumstances. Yeah. I mean, the first thing is I would say that this cosmetic gynecologist, if he really wants to do this as a scientific endeavor, he needs to get more cadavers, number one, and locate this spot repeatedly in multiple individuals. And secondarily, yeah, to find somebody who can do some neurophysiology and actually try and stimulate nerve cells that are supposedly involved in the G-spot. I mean, part of this, I mean, there are people who, looking at the media coverage of this story, there are people within the science media who are saying, oh, you know, it's terrible journalism that the story is even framed with the question, does the G-spot exist? And
Correct.
I don't think that's a terrible question. Sure, we want to know. I mean, it's the same thing as, you know, do people sense pheromones? Do we have a vomeronasal organ? Still not something that's been located in people. So there are things we don't know about ourselves that would be great to find out. And there's enough anecdotal evidence from people talking about orgasms and G-spots that it would suggest that it might exist, but maybe it's not a single structure. Maybe it's made of a number of sensitive spots. Maybe it's not the same on every individual. Maybe not everybody has one. You know, so it all comes back to me, to this idea of it's one 83-year-old woman's cadaver. That's it.
And I would expect it would be absolutely different in every woman. Every woman's lady parts are different. Does that make sense? That makes sense,
Yes.
right? You understand without
Yeah.
going into any detail whatsoever.
Yes.
All lady parts are
Individuals
different
are
and distinct
unique.
from, yes.
Individuals are unique. Yes.
I just want to make sure I communicated that without, you know, leaving it too vacuous there. Okay.
I don't think it was vacuous at all. All right, let's move up from finding the G-spot to trying to how about curing autism? Kind of.
That would be pretty intense and awesome. What do you got?
Yeah. And okay, I have two really, really interesting studies that came out this week. One is an agent that's been, this is once again curing or reducing autism-like behaviors in mice. So it's not a human study, but there is a chemical agent, a compound called GRN529. And this agent, when injected into mice that are bred, they're BTBR mice. They're genetically bred to have autism-like behaviors. So antisocial, what are some repetitive grooming behavior, other related behaviors, that when given this compound, the autistic-like behaviors were decreased to a point that the animals were almost normal. And so the interesting thing about this study is that the GRN529 is part of this class of agents. They inhibit activity of a type of receptor protein on neurons for glutamate. And so there are other agents within this class that are already being tested for patients with autism-related syndromes. So this class of agents, this class of chemicals is already being used and being tested to treat autistic-like behaviors. And in mice, this one particular agent improves behaviors to make the mice more normal. And it's just an amazing idea. The idea, it would be very easy for this compound, if it were to work in people, to be safe in people. Even they think since it's already being tested, other agents, similar agents are being tested, that it will be safe in people, that this would be a way to reduce autistic symptoms in people with, a daily pill, like just a pill or a daily injection, something that would affect the brain cells by just blocking this receptor, blocking the ability of the brain cells to respond to glutamate. And if we think of a lot of autistic spectrum disorders as related to not necessarily the miswiring of neurons, but neurons responding incorrectly to chemical signals. And if you can improve the way the neurons respond, then you're pretty much treating the disease. So this is really, really interesting research, and it just is fascinating to me. So I look forward to hearing a lot more about this later. Then there's... Oh, go ahead. Sorry.
Just real quick, two quick things too, because there was another study which I didn't bring tonight that possibly is linking some cases of autism to bisphenol A exposure. But one of the most, somebody says, well, great, they've come up with a cure that you have to go through a pharmaceutical company instead of removing the cause from the environment. Autism is strongly linked to genes, and it's it's a genetic disorder mostly, which maybe can get triggered by the environment a bit, but mostly it's genetic. And one of the things that we've seen in phenomenons where there's areas where there's spikes, for instance, Palo Alto is an example. The other end of the gene that's affected that causes autism, the other end of the spectrum of the same gene being present, creates a really high functioning ability to manage information. So you have an area where you have a lot of tech and computer programmers, people who can handle manipulating large amounts of information gathered together in one place. It's very logical that the gene expression, which can go either direction, is going to show up there more frequently than it does other places. So
and additionally, like recently it was shown that it's suggested recently that men, as they get older, experience a higher mutation rate within their sperm. And so older men are more likely to pass on genes that will lead to autistic syndrome, autism spectrum disorders. So in the high tech places, or someplace like Silicon Valley, you have people who are focusing on their careers, not necessarily focusing on having kids until later in life, and that's an additional factor that could go into it.
Right. So there's lots of factors. Environment is probably one of the weaker factors of the bunch at this point, except for vaccinations. We do have good information that when they removed mercury from vaccinations, autism rates actually increased, and therefore mercury in vaccinations was suppressing autism in society, and therefore we need to put more mercury into just to make an ultimate mockery of that whole correlation. They removed it and the rates went up. Okay, now what? Oh, well, mercury's good for children. No, you were wrong in the beginning. You're wrong now.
Yeah, but I find this a very fascinating area of research, and it's the fact that it's so specific. It's targeted at blocking if neurons are firing too much, like glutamate is excitatory, and so if you have too much neuronal excitement, and so the idea that autistic brains can't deal with a lot of stimulation because there's already a lot of stimulation going on, that if you could reduce that, that the baseline level of activity in the brain could get to a more manageable level. It's just fascinating. I really, really do hope to hear more from this.
Yeah, and one last point that just needs to be covered. Didman says, so explain how this sudden genetic expression is, for increased autism occurred so quickly. It may not have occurred quickly at all. It may be that, I mean, this wasn't autism wasn't a diagnosed disease in the way it is today. You would have likely just thrown it into a category, a very large generalized category of mental retardation or schizophrenia, which was a very broad. I mean, that which we call schizophrenia gets sliced down further and further each year, and we get new, more targeted diagnoses as we learn more. So
yeah, that's part of it. It's not all of it for sure. Misdiagnosis in the past, there is more
no
diagnosis or
earlier.
no diagnosis. Yeah. That's part of it, but it's not the whole story. There is something else going on, but we're trying to find out. And like you said, there's a very high genetic component. And it's a spectrum disorder. So it's like something, it's like cancer. It's a category of disorders where there's just a lot of stuff going on. And there are many different incarnations for many different genetic and environmental reasons, combined in different ways. And it's so in coming to, one solution, one ring to rule them all, it's not an easy thing to figure out.
Right. And it gets to the point where you can make correlations with just about anything else that's spiked in our society.
Yeah.
So that information is totally useless.
It's the chemicals. It's the nanotechnology.
It's watching too much television. It's the increase in cable. It's increasing those smart meters. I mean, it goes on and on and on.
It's cell phone use.
Right. Oh, it's cell phones now. Yeah, it could go on
entirely
forever, right?
Yeah, it can. So I hope that we just
wait for science to tell you what is true. Do not come to your own conclusion.
Yes, wait for science. Unfortunately, sometimes science moves too slowly for people. But anyway, do you have another story there in that pile of stories?
Oh, absolutely. What do you want? Your God on brains or mad cow cure? I'm going with the mad cow cure first. This is prions in the brain eliminated by homing molecules. Toxic prions in the brain can be detected with self-illuminating polymers. The originators of the study at Linköping University in Sweden have shown that the same molecules that do the illumination can also render the prions harmless and potentially cure many fatal nerve-destroying illnesses, including mad cow disease. The researchers and their colleagues at the University Hospital in Zurich tested luminescent conjugated polymers, or LCPs, on tissue sections from the brain of mice that had been infected with prions. Results show the number of prions, as well as their toxicity and infectability, decreased drastically. This is the first time anyone has been able to demonstrate the possibility even of treating illnesses such as mad cow and Creutzfeldt-Jacobs with anything, let alone an LCP molecule. When we see this effect on prion infections, we believe the same approach could work on Alzheimer's disease as well, says Peter Nielsen, researcher at the Bioorganic Chemistry funded by ERC, European Research Council. So this is a tremendous step forward. One of the things, though, I know this is a very... the sort of drawback of even the fact that you found a cure is the fact that, like the Creutzfeldt, like the mad cow, that happens quickly, very quickly, because as soon as these proteins in the brain go rogue, they have a magnitude of reproducing themselves and creating the serious brain damage. Death then follows very quickly. So this is the sort of thing that, if it got to the point of a cure, we'd have to identify it and apply it very quickly. Because the course of the illness is relentless, and when prions fall to pieces and replicate at an exponential rate. When researchers inserted LCP molecules into their model system, the replication was arrested, which is possible through stabilizing the prion aggregates.
Yeah, what's interesting about the prions is that they're contagious. So if you have a misfolded, they're little proteins, and if you have, and they're not always bad. It can be okay. But if they misfold, then they kind of become sticky. And so they will bump into another prion protein and kind of stick to it. And so you have a bunch of these prions that kind of form together and they clump together and they form aggregates. And the original misfolded protein will cause misfolding in the other proteins. And so then you have this gumming up of whatever the prions were supposed to be doing in the first place within the body. And things stop working well, and you have the disorders that we know as mad cow or Creutzfeldt-Jacobs. And so if you can stop those aggregates from building up, if you can stop the transmission of the misfolding process, then you can completely block it if you catch it in time. Which it sounds like that's what they're doing.
It sounds like it may have, I didn't get into how they did this study in the first place, but it almost sounds as though it were one of those wonderful, slightly accidental, right? Because they were using a self-illuminating polymer. Hopefully, I think the idea being then you could track the motion of prions and you can sort of see what's going on. But they ruined their experiment because this illuminating polymer shut down the prion aggregates. So then they're like, oh, we got to redo the research. Every time we do this, it fails. And then somebody's like, yeah, but wouldn't that be really great if you could do that in real active brains that are been infected? Oh yeah, that's what we have here. It's not a failed experiment. This is a cure for a disease. Wow. Yeah, they're gonna do they're gonna do some further tests. Gonna try to see if they can repeat this. They're gonna do this on fruit flies next to see if they can. They have fruit flies with an Alzheimer's-like nerve disorder. They're gonna see if they can have the same sort of effect there. Amazing.
Oh, that'll be interesting.
Amazing that the tool you're using to attempt to do the research turns out to be the answer right there. That's brilliant. Brilliant science. I've got one more to go. How many? You got another story, Kiki?
I have one more to go, but yeah, let's wrap it up here pretty quickly. My last story for the night is a world robot domination story.
Dun, 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 dun.
That's right. And according to a recent analysis of the computers at the educational testing service that grade your SATs and how well they grade essays, it turns out that they perform just about as well as people. So the essay that you're writing for the SAT, very likely that it's not being graded by a person, but by a computer, and that the computer is doing just a fine job at it. So all those grading jobs that we once could have paid teachers to do, just computers, robots. The robots are grading your tests.
And there's a good reason for it too. I mean, the excuse, or it's a great excuse for a teacher or TA not having to be involved, which is that what they're looking for is the cheaters, and it almost doesn't matter whether or not your work is good, poignant, that you made a good case for an argument, but if they find that huge chunks of your text are in this sort of database that you've borrowed from of homework or whatever, then they know that you've just copy and pasted your work and didn't actually do it yourself. And so the computer's designed to track down those cheats.
Yeah. So this particular study, it's called Contrasting State-of-the-Art Automated Scoring of Essays Analysis. Comes out of the University of Akron. And it compared the results from 9 automated essay scoring engines on 8 essay scoring prompts drawn from 6 states that annually administer high-stakes writing assessments. Student essays from each state were divided into 3 sets: training, a test set for a blind, and a validation set. And the essays were from the grades 7, 8, and 10. Total sample size of essays was 22,029. So when we were talking about a sample size of 1 earlier, not very, so not a sample size of 1, not very conclusive. Sample size of 22,000. Doing all right.
That's a good sample size.
Yeah, so the accuracy rates were computed. So the 6 of the 8 essays were transcribed from their handwritten responses, and the accuracy rates were computed at about 99%. And they found that the results overall showed that automated essay scoring was capable of producing scores similar to human scores for extended response writing. I mean, basically, because there are certain formulas to writing an essay for something like the SAT. You want to have your thesis statement. You want to have proper grammar. You want to have supporting sentences, conclusion sentences. You know, is it 5 paragraph? Is your essay 5 paragraphs long? There's a particular formula, and if you fit that formula, you're very likely to hit all of the marks that both a human grader and a computer grader would be going for.
Right. And having seen a plethora of those essays in the past, while others were grading them, I can understand that it is almost an act of cruelty to make anybody read an entire class of 33, 34, 40 students writing the same paper and trying to BS their way through it. It's brutal what you'd have to sit through to actually grade them by hand. I'm kind of thankful the robot has taken over that.
I know. This is one of those jobs that maybe that's okay. All right, but there have been other studies looking at computers versus humans, and other studies have not shown a similar result. So once again, this is only one study, and there are people at MIT, very brilliant people, who make it part of their job to try and fool the computers to show that the computers aren't so good. So, the computers, the robots, they don't win yet on this one. Okay, what about God, Justin?
We're pretty much out of time, but I'll just snip it out. University of British Columbia study finds that analytic thinking can decrease religious belief, even in devout believers. This is a study that was just published in the journal Science. It finds that thinking analytically increases disbelief amongst both believers and skeptics alike, shedding important new light on the psychology of religious belief. So basically, they had somebody sit down, do a task that required a lot of analytical thinking, then rated them on their beliefs. And those who didn't have to sit down and do an analytical thinking first were largely more on the belief side. Those who were forced to do a bunch of analytical thinking first, much less so. Interesting. But it's one of those things, too, one of those stories that sort of sounds like what you would expect somehow.
Yeah, it's not as simple as you thought.
But it might be, yeah.
Yeah. Oh, and for people in the chat room who were debating the pronunciation of prion, it can be pronounced either way. Prion or prion. So with that, we will end the show, and we will be back here next week with more science news. Justin,
thank you, everybody, for listening. We hope you enjoyed the show. Twis is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have an Android device, you can look for Twis for Droid. That's Twis, the number 4 Droid, in the Android marketplace. And we also exist on the iPhone marketplace if you happen to have one of those devices.
And for more information on anything that you have heard here today, show notes will be available on our website, twis.org. We also want to hear from you, so email us. I'm Kirsten at thisweekinscience.com, and Justin is Justin at thisweekinscience.com.
Be sure to put Twis, T-W-I-S, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also contact us on the Twitter, at Dr. Kiki, at Jacksonfly, at Blair's Menagerie. And we also have a Twis Science. Twis Science is also. We have more Twitter handles than we
Twisians.
have people at this point involved in the show. There's also, if there's a topic you would like us to cover or address, a subject for an interview, an interesting haiku, please let us know.
And we are going to be back here, like I said, next week. And we hope that you'll join us once again for more great science news.
And if you've learned anything from today's show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And this week in science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma. Got the eye. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science.